Our next speaker is Beatriz Diez from Pontificia Universidad Católica de Chile, and she will be talking about microbiome structure, biogeography, and interactions in hot spring environments. Go ahead, Bea. You are muted.
Okay, thank you very much, Rodrigo, for your introduction. And I cannot go back. Let's see. Can you see the whole complete screen?
Yeah.
Okay.
Yes.
Perfect. So thank you so much for giving me the opportunity to be here today. Try to show you some of the work that we have been doing in the last years, basically the last decade already. Time passed really fast. Related to the microbial ecology of in hot springs environments. First of all, I want to thank basically all the funding that has been helping us during all this decade. Basically the National Agency of Research here in Chile, and also the Iniciativa Científica Milenio with the Millennium Institute Center for Genome Regulation and the FONDAP Center for Climate and Resilience Research. And we all know that, and it has been proved some time ago, that microbial diversity and species distribution has a dependency of temperature in many natural systems. And it has been proved, for instance, with the global ocean microbiome, and also with the at a continental scale of microbes, diversity of microbes in forest soil. But if there is a system where we believe that temperature is important as a driver for species distribution, are terrestrial hot springs. They have like a wide physicochemical gradient over short distance, which make these systems ideal island models for identify different conditions, including temperature, but also biotic interactions that drive community changes in not only the large, but also small scales. They are also ideal models for studying microbial biogeography and endemic patterns, and assess the roles of thermophilic communities in related to biochemical cycles. Also, they are important to evaluate the response to environmental changes and their potential relevance in adaptation and evolution under different scenarios like the actual climate change crisis. So where are the hottest non-geothermal regions in the world? Well, they are basically all over the world, but there is a region that is called the Pacific Ring of Fire, where there are a lot of volcanisms, and then the occurrence of many hot springs, terrestrial, but also oceanic hot springs all over this ring of fire. And actually, Chile is sit in this ring of fire, and that's why we, from north to south in Chile, in our geography, we have like a lot of occurrence of hot springs all over our geography from north to south, and it's related to volcanoes because we have more than 80 active volcanoes at the moment. So we have been studying in my group several of these hot springs, like the most emblematic one in El Tatio in the north part of our country in the Atacama Desert, which is actually the third geothermal field bigger in the world and the first in Latin America. In my group, then, we have been studied the effect of both abiotic and biotic factors and geographic distance on the structure, function, and interactions of microbial communities at different levels, scales, like from local to regional and latitudinal, and finally global scale. Starting with the local, we have a PET hot spring, which is located in the northern part of the Patagonia, which is called Porcelana, and that the one that has been our model system because it has a neutral pH and the occurrence of thermophilic photoautotropic mats, which are very important for us because they are ideal model system for studying the principles of microbial ecology, as they are like a small scale, closed, self-sustained benthic ecosystem that comprise the main element cycles, trophic levels, and food webs, as the one that you can see here, that in only 2 or 3 centimeters, all the transformation and exchange of gases and nutrients occurs in a really short and not that much complex microbial community. So for several years now, we have been studying these communities, and in particular, trying to understand their contribution to the biochemical cycles like this carbon and nitrogen through investigating how they fix and assimilate compounds related to these biochemical cycles. And we did that using different methodology like stable isotopes to get rates, also enzymatic activity like reduction acetylene for trying to understand more about the activity related to nitrogen fixation, and the use of expression of different genes related with all these assimilation and fixation compounds. Through that, we have proof that many of the primary producers that work in these microbial communities are very important as contributors to the carbon and nitrogen fixation and assimilation, like cyanobacteria. For instance, the ones related to the genus Fischerella, and in particular the species Fischerella thermalis. We also have been working to know more about the functional capacities and the transcriptional activity of all these photoautotropic mats and the microbes that contribute to these systems from a thermal gradient, where we can also demonstrate that there is a temperature dependency related to functions and not only to the occurrence of certain groups of organisms. We also did prove through metabarcoding 16S, using the 16S gene diversity, show that there is a temperature dependency that generates a niche partitioning in the thermal gradient of some of these hot springs like Porcelana in the northern Patagonia. As you can see here, we found around 58 degrees threshold temperature, where there is a turnover change between 2 primary producers in these microbial mats, which are cyanobacteria, below 58 degrees, and then it changed the abundance of chloroflexota, another primary producer that is working in the system, and then probably there is a functional redundancy of these 2 group of organisms that behave to maintain all the functions, the more relevant functions of primary producers in these kind of systems. We also wanted to prove that in a more global scale, and then we move to mine more than 100 metagenomes that were available in the databases together with our own metagenomes to try to understand more about a global scale of taxonomic and functional organisms that work in these hot springs all over the world. From that, we did also mine 16S genes diversity, and again, we could prove that there is a niche partitioning that occurs also across the temperature gradient at a global scale. First, we again found this 58 degrees threshold that I showed you before in a local scale, but then we also found through or thanks to this bigger amount of samples with a bigger range temperature, that at 70 and at 80 degrees threshold, they also occur a turnover of a species between bacteria, thermophilic and hyperthermophilic, defined by these 70 degrees, and also on 80 degrees when we separate hyperthermophilic bacteria from hyperthermophilic archaea. Also, thanks to this mining of metagenomes all over the world, is that we managed to understand a little bit more about taxonomy and functional novelty through more than 4,000 genomes that we managed to assemble from metagenomes, which reveal also niche preferences in these terrestrial hot springs all over the world. Basically, more than 80% of all these new MACs were classified as potential new species, and this MAC contribute to most of the poorly described phyla that we have in our databases. So that was very interesting to get new MACs that will help us in the coming future to understand more about functional capacities and phylogenomics into the thermophilic group of bacteria and archaea. In particular, we are cyano lovers in my lab, so that's why we were doing some more specific work in MACs related with cyanobacteria that we managed to get from these metagenomes. And then we did phylogenomics and classification of these new MACs in the GTDBTK platform. And then we managed to see that many of these new MACs recovered from these metagenomes are actually new species, even new genus, and even some new families and probably new orders. But what is more important for us is that we could do, now that we have more genomes, genomic comparisons between thermophilic genomes against the non-thermal relatives genomes. And we found that hot spring genomes are smaller and with higher GC content. Their proteins are shorter and more hydrophobic. One very interesting thing is that they have more frequency of CRISPR-Cas defense systems. That is something that is not new. It has been proved before for other bacteria in culture. But what we wanted to know more, it was a more global scale, how important are these defense systems in hot spring. And then what we did is just mine these more than 4,000 new MACs that we recovered from these more than 100 metagenomes, and try to recover all known defense systems that we know until now in this from natural systems and cultures. And then we found that definitely CRISPR-Cas are the most frequent defense systems in hot spring, in terrestrial hot springs, very much followed by restriction modification defense systems. And when we compare these two type of defense systems, we see that the more the temperature increase, the more increase the frequency of CRISPR-Cas defense systems. That suggests for a temperature dependency and a potential strong phage-host interaction by, for instance, coevolution between the viral viruses and their host in this hot spring. As we wanted to know more about these CRISPR-Cas defense systems, they are very important in hot springs. We mined these metagenomes trying to look for Cas1, which is the more conserved enzyme in the CRISPR-Cas, most used, also enzyme used for phylogenetics in CRISPR-Cas systems, and try to follow by geographic regions. And what we found, it's very interesting, is that the Cas1 CRISPR-Cas enzyme was very specific in terms of diversity, followed by their location, geographic location, which actually suggests for a potential endemism in the pair interactions phage host. So that brings us to try to understand more about this interaction of viral communities and their host in hot springs, both at local but also at global scale. And as you can see here, we use metagenomes from different continents, countries, and different hot springs all over the world, using not only microbial mats, but also other habitats like sediments and waters. And what we found is that a great taxonomic novelty of viral genera related to hot springs. Basically, at the beginning, we tried with different methodologies, and we were failed so much to assign taxonomically to the viral community. Then we moved to the global viral protein cluster networking for getting more into the knowledge of the taxonomy of these viral communities. And as you can see here, less than 10% of the viral genomes that we recovered from all these hot springs were assigned to known viruses in the databases. And they were related basically to Caudoviricetes, which are viruses that infect bacteria and archaea. So it means that 90%, more than 90% sometimes, only of these viral communities only cluster between hot springs, which suggests a restriction to these environments. When we try to see how similar or dissimilar were these communities between hot springs around the world, we did an analysis of beta diversity. And as you can see here, they differ very much between different geographic regions in their distribution, which actually suggests for a limited global dispersion, but potential local and regional adaptation. So that was very important and interesting for us. And that's why in the last years, we are working more specifically in viral communities on a local scale in El Tatio geothermal field in Atacama, here in Chile, because this geothermal field has a great geographic isolation and uniqueness in terms of conditions that may select for this high amount of endemic local viral communities. I'm not going to go through what we found because I know that I passed the time, but I just let this slide for you to be motivated to come to Chile and visit some of our main microbial systems, natural systems, like El Tatio in Atacama, and many other environments where microbes are very important in these systems and can help us to understand more about adaptation and evolution in our planet. And I want to thank you, my students and my main collaborators here in Chile, but also all over the world, supporting my work. So, thank you very much for your attention.
Thank you very much, Beatriz.